Hey you guys, welcome to 16.1. We are talking about inheritance, and chapter 16 is about DNA replication, but 16.1 is going to be about the discovery of DNA. So let's get started. We finished our last chapter understanding that T.H. Morgan and Stewart had determined that genes exist on a chromosome, but they didn't really understand what a chromosome was at the time. They knew that there was a molecule that was passed on from generation to the next, and they called it the transforming molecule. It was able to transform the traits of whoever it was given to. And they thought at this time, they narrowed it down, scientists had narrowed it down between proteins and nucleic acids. Most scientists thought, though, that it was proteins because proteins are so diverse. There's so many different proteins that have so many different functions. They thought that this was going to be the molecule that was able to give so many different traits to so many different types of organisms. Nucleic acids, the only variability in nucleic acids is the A's, the T's, the C's, and the G's. And scientists thought, man, there's no way that just A's, T's, C's, and G's, just four different changes in this one molecule could possibly be what caused all of the changes in life or all the variation in life. First experiments done in 1928 that showed that there was a molecule, this transforming molecule, that could change an organism's genotype and phenotype was done by Frederick Griffith in 1928. He was trying to create a vaccine for streptococcus pneumonia, or for pneumonia. And what he did was he took living, smooth, pathogenic cells or bacteria, and he injected it into a mouse, and the mouse died. So let me grab my draw tool here. Blue is perfect, and let's draw a smooth cell. And that should make sense, right? So he injected a mouse with pathogenic bacteria, and the mouse died. Then he injected another mouse with a rough, non-pathogenic cell. So here's our rough cell, non-pathogenic. And he injected that into a mouse, and the mouse lived and was fine, which makes sense. It was yet another control. Here's another control. He took heat-killed S cells. So he took this cell right here, and he heat-killed it. So it's pathogenic, but then he killed it, so it's no longer pathogenic. And when he injected it into the mouse lived. It was fine because the bacteria was dead. Finally, his experiment, the mixture of heat-killed S cells. So he took this heat-killed S cell, so let's draw the smooth cell, which is pathogenic, but it's been killed off, and he mixed it with the rough cell, the living non-pathogenic cell. When he injected that into the mouse, counterintuitively, the mouse died, which didn't make sense because the pathogenic bacteria was killed and it was the non-pathogenic bacteria that was still living. And so what this showed was that the pathogenic bacteria was able to pass on something, a transforming molecule, that to the rough non-pathogenic bacteria, which changed not only its genotype but its phenotype. This cell became smooth and pathogenic because of the transforming molecule, or because of the heritable molecule that was passed on from one bacteria to another. So this showed that there was something, a heritable trait or a heritable molecule that was being passed on. Let me just turn this draw tool off and we can move on to our next slide. Now the hunt for what that molecule is. They said, so we knew it was either going to be a protein or a bacteria. And Hershey and Chase designed an elegant experiment using a bacteriophage. In fact, it was a T2 bacteriophage. And a bacteriophage literally is a bacteria eater, or it kills bacteria. And a bacteriophage is composed very simply of two things: a protein coat, so the head of this phage right here is a protein coat, and a nucleic acid in the center of it. And so these are the two types of molecules that scientists were arguing about whether it was, which one was the transforming molecule. And what the scientists did, what Hershey and Chase did, let me move this up, and they made a radioactive label for, let me go to this picture, I drew it. They radioactively labeled the protein coat, so the head here, they radioactively labeled sulfur, so they could track the sulfur. So if the sulfur was moved to somewhere else, they'd be able to follow it. And then the genetic material, the nucleic acid, they labeled the phosphorus. They chose these two things because proteins don't have phosphorus and DNA, or nucleic acids, don't have sulfur. So they were able to track what was going to be passed on. What they did was they, in a solution, they put bacteriophages and E. coli, and they allowed the bacteria phage to attack or to inject, to attach onto the bacteria and inject the transforming molecule, or the molecule of heredity, into the bacteria. And so here, whatever it was, whatever that molecule of heredity was inside the bacteria. And they wanted to know, was it protein? Was there going to be sulfur in there, radioactively labeled sulfur? Or was it the nucleic acid? So was there going to be a radioactive phosphorus in there? What they did, they had to first agitate the solution to shake the viruses off of the bacteria. And then they put the solution that had phages and bacteria into a centrifuge. They let it spin for a while, and because bacteria are more dense, they formed a pellet at the bottom, and the supernatant was filled with the less dense phages, bacteriophages. So whatever was in this pellet was going to indicate what was the transforming molecule. So if the phages are up here, then whatever they injected into the bacteria, because the bacteria was down here, was going to indicate what it was. And so, of course, they found that it was radioactively labeled phosphorus. And then to double check their work, they checked the supernatant, and they found that, yes, the supernatant was filled with radioactively labeled sulfur. And that was evidence that the transforming molecule was, in fact, the DNA. And it was the A's, T's, C's, and G's that incredibly code for all of the genetic variation in all of life. So now that we know what molecule it is, scientists wanted to start identifying its composition. How was it made? And one discovery was made by Erwin Chargaff in 1950. So he analyzed the composition of the nitrogenous bases in the DNA. And what he found was that there is molecular diversity among different organisms. So he saw that, for example, a sea urchin contained 30.4% adenine, whereas humans have 24.7. And so a horse is going to have a different amount of adenine than, let's say, a tree or a carrot. And this is because our genetic code, the amount of A's, T's, C's, and G's in each of our genome is variable. And so it showed that there was molecular diversity that coded for our variation in life. He also saw that there was equal amounts of adenine and thymine. So they had similar percentages, whereas guanine was similar to cytosine. And so he was starting to make the connection that the two were going to be, eventually found that they were paired up. He didn't know that they were paired up exactly, but he knew that there were even amounts of adenine and thymine, guanine and cytosine. Another discovery by Rosalind Franklin, she used X-ray crystallography. So what that is she used a crystallized structure of DNA and sprayed it with X-rays. And the X-rays bounce off the crystalline structure and create a pattern. And this is the pattern that it created right here. And while it's nonsense to us, it showed her that DNA was actually, or is actually, a two-stranded molecule. Many scientists at the time believed that it was a three-stranded molecule, but she had proof that it was a two-stranded molecule. And this image also showed that the strand makes a full turn every 10 bases. So right here, one full turn. And that each base pair is 0.34 nanometers apart. So between these bases, it's 0.34 nanometers apart. So this is a huge discovery about the structure of DNA. It wasn't the final discovery, but it was a big help. And the people that used it to help them make their discovery was Watson and Crick. So they used the discoveries of previous scientists by piecing the molecules together like a puzzle. So they used Chargaff's discovery that that there's a similar amount of A and T and G and C. And they used Franklin's image to discover the actual complementary base pair and the actual structure of DNA. And so they realized that there's a uniform distance between the two sides, and that the pyrimidines are going to have one ring. So thymine and cytosine are pyrimidines. I remember this because it's a longer molecule, but it's a longer word, but a shorter molecule. And that the purine is a shorter word, but it's a longer molecule because it has two of the bases. And so a pyrimidine is always paired with a purine. And so that's going to add one ring to two rings, so it's going to have a uniform 3 rings down the entire structure. When Watson and Crick published their work in Nature magazine, they noted that it had not escaped their notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for genetic material. They suggested that the DNA was going to split open and be read and copied and then closed back up. We call this the semi-conservative model, but we weren't sure. And so the next part of 16.2 is going to start talking about the different types of theories on DNA replication. I hope this helps, and I'll talk to you guys later.